Welcome back everyone. Today we are diving into lecture 3 of our course, Bioinformatics and Application. I'm Dr. Asela Wijeratne and we will explore how DNA sequencing is revolutionizing research and medicine. Our goals today are to understand how Sanger sequencing works, how to interpret sequencing data, and quality score, and how to prepare sequencing data for analysis. These skills are foundational for bioinformatics. DNA sequencing has many uses, from diagnosing genetic disorders in medicine to trace human migration in anthropology. It is also vital in forensics, agriculture, and ecology. Think of one example for each to connect theory with real-world impacts. There are 3 generations of sequencing methods. Today we will focus on Sanger sequencing, the first generation method. We will also talk about the second generation and the third generation long read technologies later in the course. Let's talk how we perform the sequencing. To perform Sanger sequencing, you will need DNA template, a primer, DNA polymerases, and mix of normal and chain terminating nucleotide. These components work together to generate a readable DNA fragment. Before sequencing, DNA must be amplified either by PCR or cloning. Then it's purified to remove salts and primers. Then you need to accurately quantify DNA before you can send it for sequencing. Too much or too little DNA can affect the results. Okay, let's talk about the primers for sequencing. You only need one primer for sequencing, and the primers are short DNA sequences that bind near the region that you want to sequence. They must be complementary to the flanking region and are typically 15 to 13 nucleotide in length. Let's walk through how Sanger sequencing works step by step. We start with the DNA and primer that binds to a specific region of the template. Then we add a DNA polymerase, the enzyme responsible for copying DNA, along with a mixture of normal nucleotides called DNTPs and a chain terminating nucleotide known as DDNTPs. Here's the clever part. Each DDNTP is tagged with a different fluorescence dye, and they are added at a lower concentration, about 1:100 ratio compared to the regular DNTP. As the polymerase copies the DNA, it occasionally incorporates DDNTP instead of a DNTP. When that happens, the chain stops growing. This results in a collection of DNA fragments, each ending with a fluorescently labeled DDNTP. Because DDNTPs are randomly incorporated, we get fragments of varying length, each terminated at a different position of the sequence, as shown in this diagram. These fragments are then separated by size using capillary electrophoresis, and a detector reads the fluorescence tag to determine which base is at the end of the fragment. This is a newer version of the method where each DDNTP is labeled with a different fluorescence label. The labeled fragments are separated using capillary electrophoresis and detected, producing a pattern that corresponds to the DNA sequence. When a sequencing run is completed, the machine produces what we call trace file. This is an example of a trace file. These files contain raw signal data of fluorescence dye signals that corresponds to each nucleotide incorporated during the sequencing reaction. Each lane, these are different lanes in different columns. Each lane is correspond to a different sequencing reaction. Now these trace files are analyzed by a software to generate what we call a chromatogram. That's a visual representation of the DNA sequencing. The peak in the chromatogram correspond to different nucleotide bases, A, T, G, C. The height and the shape of the peak help us to determine the confidence of the base call. Let's talk about what you really receive from sequencing facility. You will typically get two different files, a FASTA file and a chromatogram file. The FASTA file contains nucleotide sequences, just the letters A, T, G, C. It's clean, simple, and easy to use for downstream analysis. But here's the catch. It's generated by an automated base caller. Now, the base caller is a piece of software that interprets the raw signals data from the sequencing machine. It looks at the peaks in the chromatogram and decides which base is most likely at that position. But, and this is important, it is not perfect. Sometimes the software misses subtle, ambiguous peaks. These could be due to background noise, overlapping signals, or weak fluorescence. As a result, FASTA file might not be the most accurate. That's why it is a bad idea to rely solely on FASTA files. You need to inspect the chromatogram, the visual output from the sequencing run. It shows you the actual dye signals, so you can verify whether the base calls make sense. In short, the FASTA file is convenient, but the chromatogram is the truth. Always double check your data before moving forward. Okay, let's talk about how to examine a chromatogram. Typically, chromatograms can be messy at the beginning and at the end. Early on, the reaction hasn't stabilized. Later, the signal fades. You can also sometimes find background noise that can interfere with the base calling. We will learn more about how to spot these issues during the class. As I mentioned earlier, base calling is often done by a software like Phred. It analyzes peak, height, shape, and background noises. If the software cannot confidently call a base, it assigns an N. We'll explore how accuracy is determined in the next slide. Phred score quantifies base call accuracy. A score of 20 means it's about 99% accurate. Phred score 30 means it's about 99.9% accurate. These scores are logarithmic. Higher scores mean exponentially lower error rate. Understanding these scores is key to data quality. If you have only one sequence, your sample size is 1, or in other words, n equals 1. To improve reliability, sequence from both ends or use replicates. This helps confirm your results and reduce error. Let's wrap up today's session by reviewing what we have learned. We started by exploring the different types of sequencing methods, from first generation Sanger sequencing to next generation and third generation technologies. We walked through how Sanger sequencing worked. DNA polymerases copies the template strand, and when dideoxynucleotide or DDNTP is incorporated, it terminates the chain. Because the DDNTPs are added at a lower ratio compared to regular nucleotides, we end up with a mixture of fragments of varying lengths, each ending with a fluorescently labeled base. These fragments are separated and recoded to reconstruct the DNA sequence. Then we shifted to sequence data analysis. We learned how sequencing machines produce trace files, which are analyzed to generate chromatograms, a visual representation of the DNA sequence. These chromatograms are essential for verifying base calls and identifying errors. We also discussed quality scores, especially Phred score, which tells us how confident we can be in each base call. A score of 20 means 99% accuracy, while a score of 30 means 99.9% accuracy. These scores help us to decide which part of the sequences are reliable enough for downstream analysis. Finally, we emphasized the importance of not relying solely on FASTA files. Automated base callers can miss subtle peaks, or always inspect the chromatograms to ensure your data is clean and accurate. Great job today. These are foundational skills in bioinformatics, and mastering them will serve you well in both research and clinical application.